Okay, so thanks for pointing that out. The class that we're in right now, which is our lab class, is 1108. And when we meet downstairs for lecture, that one's called 1308. So it is actually two different classes. And just as a side note, when you see it show up on your SOS transcript, it will be there as two separate classes, Biology 1308 and your grade, and Biology 1108 and your grade. They will be the same grade because we calculate your grade based on both things together. So whatever you're seeing on your blackboard as your current average, your weighted average, that's what's going to show up for both your 1308 and your 1108. Because I consider it one big class for grade calculations. Okay? But that's a great thing to point out. The reason we did that is because especially non-majors are losing that 1 hour class when they transfer it. It was a 4-hour class, and when you transfer it, they just give you credit for 3 hours of science, and you lost that other hour. Well, now you can pull that other hour and put it in your electives. So it might work out better for you for getting credit for everything you transfer. Yes, ma'am?
So when you transfer only 3 hours, will transfer
for a science.
And then the other one you can put for an elective.
Towards an elective. Yeah. So a non-major, I don't know if you've looked at the state of Texas, but for non-majors, you're only allotted 6 hours of credit for science, which is 2 classes. But saying, Jack, you're going to get 8 hours. So we pulled those extra hours out so you can put them in your electives when you transfer. But okay with that? That's for your baccalaureate degree, your BS or BA degrees. So that's why we did that. Because if you were here last year, you know that we didn't have it separated into 2 classes, but now we do, and that's why they told me we did it. Okay, I have no idea. I just teach. Okay, so let's get going on that, what I call teaching. Okay, so exercise 5 was enzymes. We're supposed to identify the reactants, the products, and the enzyme in a chemical reaction. Now, the chemical reaction I'd like to work with everybody is on page, actually 58. The chemical reaction I picked is actually on page 58. And you maybe already boxed this chemical reaction in your lab manual. And the reason I chose it was the chemical reaction we did use. And so I just used up here, if you'll take a look at what I did, I just used the formulas. I didn't use the words. But you can see what each thing stands for in your lab manual. Let me go over it with you. I have H2O2 is the reactant. I didn't use the word reactant. What did I use? An R. H2O2 is hydrogen peroxide. You may remember that day we had brown bottles of hydrogen peroxide on our table that day. And then everybody knows this other side. What is H2O? Oh, people, you scare me. What is H2O? Water. See why I get scared? Huh? And what is O2? Oxygen. And if you'll notice, I bracketed those and put a P on it. What does that mean? Products. Those are my products. So anything on the left or before the arrow is a reactant. Anything on the right or after the arrow is a product. Okay, now I added a little bit here. I also want you to be able to recognize an enzyme in a chemical reaction. Everybody check out my enzyme. What's my enzyme? Catalase. Now, there's 2 ways to know that was an enzyme, excluding the way that it has a little letter E above it. Because you know on a test, does everybody know on the test that light pink writing is not going to be there? Is everybody okay that that's not going to be there? Okay. So, besides that I already labeled it an enzyme, how do you know 2 ways that catalase is the enzyme? Thank you very much, Jordan. It ends with ASE. Any other way of knowing it's the enzyme? It's on top of the arrow. See, it's not really reactant and it's not really a product, so we put it on top of the arrow. So that's the 2 ways to know that's the enzyme. Everybody's okay on that? Okay, so super. All right, for number 2, define enzyme substrate, active site, and enzyme substrate complex. Well, you know, guys, we could write definitions for those, but I would rather apply our knowledge of what those mean with page 57. Everybody go back and look at page 57. Now, for page 57, there are some visuals there, and what I'd like for you to think about is, could you label those visuals with these words? Substrate, enzyme, active site, enzyme-substrate complex, if I took the words off. So what do you think? Could you label these visuals if all the words were gone? Shake your head yes or no, I don't know. Deanna said yes. Alex, Colton, could you do it? Okay. So, look, guys, I didn't pick that picture. I used a different picture. It's the one I found from your textbook. It's similar, but a little bit different. So, if you'll check it out, what I did, if you'll notice, I crossed out the word enzyme. So, let's pretend that wouldn't be there. See, all these words wouldn't be there. And what I've noticed on students is they don't have really any problems, except sometimes they confuse the enzyme and the substrate. It's the only time I can see people having problems. So I want us to brainstorm. How are we going to know this blue thing is the enzyme?
That's what it attaches to.
It's where something attaches to it. Okay, anything else? There's more. Because it's at the start. Okay, anything else? The indentions. What are these indentions called? The active site. So only the enzyme would have the indentions, which you and I know are the active site. You guys know that's the real important part of the enzyme, right? The active site. Okay, here's something I noticed. That the enzyme doesn't change. Let's look at the picture. The enzyme doesn't change. Does the substrate change?
Yes.
Yes. What does it change into? And then into? What happens to the substrate? It becomes something. This last one, what is that? Products. So substrates change into products. Substrates change into products. Now, as you're making note of that, I want to remind you, substrates are simply reactants that use an enzyme. That's all a substrate is, a reactant that uses an enzyme. Okay, so again, study page 57, page 57. Okay, on the same question I have, what is the effect of an enzyme on the energy of activation in a chemical reaction? And I did a real quick little sketch right here. Let me just go over it with you. So I have this graph, and the y-axis is over here. Do you guys remember what I use for capital E? What is capital E? Energy, and notice low energy would be down here and high energy would be up at the top of the y-axis. And then I had to squeeze it in, sorry about that, but the x-axis, can you read what the x-axis is? Time. In case you can't see it, that's time's going by. Just time is ticking away, tick-tock, tick-tock, just like right now. Okay, and you guys know what we start with is substrate, so I'm going to use an S for substrate. And what do we end with? Product. So I'm going to put a P for product. So what does S stand for on my graph? Substrate. What does P stand for? Product. Okay. I've got two lines. I don't know if you can see them. Let me point them out. I've got this one that's kind of a yellowish color. You see this one? This one's a little bit darker. Okay, and I labeled this one right here, with enzyme. How did I know that one used an enzyme?
Sped up.
It sped up. How do you know from looking at that graph it went faster? How do you know? No? You start at the same place, and you end at the same place. It has to do with this and this. What are these humps called? Very good. Haley said energy of activation. And over here on the side, I wanted to talk about this. This is called the energy of activation. E sub A is the energy of activation. It's how much energy has to go into a chemical reaction to make it go. Now I put arrows on the energy of activation. Here's the energy of activation for the lower reaction, right here. Notice how tall it is. Here's the energy of activation for the other one, the darker line. Everybody knows how tall that one is? Everybody see the difference? So let's go back to the question that's in black. What is the effect of an enzyme on the energy of activation? It lowers it. Everybody see on the graph that it lowers it. So the answer is it lowers it. It's just a little graph to remind us about that. Okay, on this question, I'm going to move forward. Don't forget, if you want to go back and see this, it'll be on YouTube tonight. Okay, number 3. What happens when an enzyme is denatured? It stops work. Yeah, so an enzyme is denatured. Something's happened to its shape. It's lost its shape, and it will stop working. We know when an enzyme is denatured, it doesn't make product, so it stops working. So when an enzyme is denatured, it stops working. It stops working. And the reason it stops working is because it changes shape. It changes shape. Now remember that we came up with 2 different scenarios that could change the shape of an enzyme, thus make it stop working. Let's talk about this first one I have right here. What does this mean with the arrow and the word temp? What does that mean? High temperatures. We're talking hot. I'm not talking warm, I'm talking hot. And you can just imagine why you got this little enzyme, which is a protein held together with hydrogen bonds, and you put it in hot water, it's going to start shaking because of all the kinetic energy. And so all of the bonds are going to start breaking, and the darn thing's just going to fall apart. So definitely high temperature will change its shape. And then look what I have here. I have 2 different arrows here. Look. What is this first arrow? I'm going over it right now. What's this first arrow? What's that supposed to mean? Low pH. What kind of pHs are low pHs? Acidic. So acids should denature enzymes. And that's true for most enzymes. Let's think of an example that's an exception in our body. Where in your body would the enzymes work at low pH? In your stomach. And there's a couple enzymes there that can do that. But most of the thousands of enzymes in your body, they do not like low pH. They do not. Okay, what about this second arrow right here? What does that mean? High pH. So give me an example of what could be high pH. Like, what does that mean? Basic. So bases will also denature an enzyme. So, okay, well, what I mean by these arrows on pH. So by and large, most enzymes would like what kind of pH? Neutral. What is a neutral pH? 7, right around 7. Does everybody know these things? Super. Moving on. All right, I got the answer for number 4 off of page 58. Everybody take a look again at page 58 and see how I figured this out. It's that chemical reaction towards the bottom of page 58. The question is, what is the function of catalase? So because it's an enzyme, I put it speeds up. Everybody agree with me that it's an enzyme, so by definition, enzymes make things go faster. So everybody okay with the speeds up part? And then I was studying this chemical reaction, and it looks to me like catalase makes hydrogen peroxide break down super fast. So I put it speeds the breakdown of hydrogen peroxide. H2O2 is hydrogen peroxide. Everybody okay on that one? Super. And then lastly, how did we judge if the reaction occurred? Bubbling. Now, I will ask you, what is the gas identity of the bubbles? Because these are not the only bubbles we've studied this section. So you're going to have to know what gas bubble we were looking at and what would be the identity of the gas bubble. Oxygen. Good job. So, do you have to know this chemical reaction? Yes. Okay, I want to move on over to question number 5. And on question number 5, I want you to make note that we're looking at page 60 in our laboratory manual for question number 5. Page 60. Specifically, the data table 5.2. Take a look at it. So you told me we're looking for bubbles. And to kind of quantify the bubbles, we did column height of bubbles. We put our tubes in the refrigerator, the incubator, and boiling water. I called the refrigerator cold up here. Cold. More specifically, I think we're right around 4 degrees Celsius. What were we at, like 3.8? What's your 3.6? So we were right around 4 degrees Celsius. Okay, warm, I think that was about 40 degrees Celsius. Yeah. And then we did not do boiling. That is way too dangerous. I think I did 90 degrees Celsius. It was still really hot though, okay? All right, so check out your data. Everybody look down at your book. When you had the cold test tube, did you get some good bubbling there? Yes? Like maybe 50 millimeters of bubbling, 45, 50, 55 millimeters of bubbling? Yes? Yes? So that's good bubbling. Okay, how about warm? How'd you do on bubbling on warm?
Okay. Denatured, but it was just 0.
On the second one?
Yeah. It was a little bubbling, not pretty bad.
I thought you guys got bubbling on number 2.
It was a higher amount.
Jordan, you didn't get any bubbling on number 2? What's wrong with you? I didn't mean that. Something's wrong with that test tube. Not you personally. I don't know. I think you're supposed to get bubbling. Everybody else got bubbling?
Yeah.
See, now you know why we do class data, huh? Yeah, there's always one. Okay, how about the really hot water? How'd you do on bubbling? No bubbling? Is everybody in agreement? No bubbling on number 3? Okay, so it looked to me like catalase did okay bubbling when it's cold, okay bubbling when it's warm, but could not bubble when it's hot. So the question will be, why couldn't it bubble when it was hot? And what's the answer? It was denatured. It just got too hot, and it denatured catalase. It made the catalase fall apart, so it couldn't function anymore. So I put why? Well, because it denatured the catalase. And you maybe have already made some notes about that on page 60 in table 5.2. I hope you already used the word denature in your book. Did you?
Yes.
Yes? Okay, it's right there.
Our numbers flipped, I did get bubbling.
Oh, okay, good. The numbers were just flipped. Okay. So there is nothing wrong with Jordan today.
Not today. Not today.
Not today. Okay, is everybody okay on number 5? Number 5. Super. Okay, earlier today for number 7, number 6 is omit. I'm sorry, number 6 is omit. We did not have time to do that activity, so we will be omitting number 6. Number 6 is omit.
Sorry, did number 5 say cold, medium?
Yeah, the words I used were cold, warm, and hot.
Thank you.
Okay, number 7, what's the effect of a pH outside the optimum range of an enzyme? So that means a pH way higher than its optimum or way lower than its optimum. And I put that it'll stop the enzyme. Why will it stop the enzyme this high or low pH? Because it causes it to denature. Diamond's correct, because a high pH or a low pH will cause the enzyme to denature. So I put that it stops the enzyme. Okay, now, I went ahead and looked at your book, and I'm looking at page 62 this time. Everybody check out page 62. Looking at page 62. Table 5.4. Table 5.4. Now, if you had to draw these 3 test tubes, just pretend we were drawing these 3 test tubes. Which of these 3 test tubes at the bottom of page 62 would you put bubbles in? Diamond is nominating number 2. Does your data support that? Okay, did you get any bubbling in number 1? No. Did you get bubbling in number 3? No. Okay, now look at their pHs. Number 2's pH. It had bubbling. What was the pH? 7. So right where we were thinking, right? Weren't you thinking 7 would be the best pH? Okay, number 1, you said no bubbling. What was the pH? 1. What's the problem? Why didn't it bubble? The pH was too low. So what did the acid in the test tube do to the enzyme? Denatured it. Okay, now look at test tube 3. Did it bubble? No. What was the pH?
Base is 14.
Whoa-wee, that is very basic. That's called whoa-wee. Very basic, okay? Yeah, wow. Okay, so what did the base do to the enzyme? It denatured it. I don't know how many times he said denatured today, but it must be an important word. Okay? So this is what I did, guys. I drew this out. You don't have to draw it out if you want to. It's all there in your notes, but I thought it might be useful for you to look at just a little visual of that, what I meant by drawing it out. So if you check it out, here's my test tube number 1, and test tube number 2, and test tube number 3. Please note test tube number 1, test tube number 2, test tube number 3 all got catalase, the enzyme, and they all got hydrogen peroxide, the substrate. So they got the enzyme, they got the substrate. So they're all set up to make the product. However, test tube number 1 got HCl. What is HCl? What is that? If you had to tell somebody that's in junior high, they said, what is HCl? Hydrochloric. It's an acid. There you go. That's an acid. HCl is an acid. Okay, and then look, this test tube got water. Number 2 got water. Everybody knows H2O is water. Okay, test tube number 3 got NaOH. What is NaOH? It's NaOH? Sodium hydroxide. You're killing me. Sodium hydroxide, is that acid, base, or neutral?
Neutral.
Really?
No base.
It's a base. Okay, so everybody check it out. HCl is an acid. H2O is just water. You're scaring me today, so I'm going to go ahead and write that down. And NaOH is a base. Now, just because of these pHs across the top, I could have figured that out anyway, right?
Yes.
Okay. I was just remembering the pHs. I think you told me. See if I got this right. Did you tell me test tube number 1 had a pH of 1?
Yes.
Everybody? Okay. Number 2 had a pH of 7?
Yes.
Okay. And number 3 had a pH of 14? Is that correct with your data? Okay. And then once again, glance up here. Every test tube got catalase. Now, I just want to comment on that. I've been reading your projects. I'm not quite finished, but I've been reading your projects. And most of you have figured out that every experiment needs a control. And that in an experiment, everything has to be the same except for one thing. And so this is a good example of that. Notice every tube is the same except for one thing. Every tube got catalase and hydrogen peroxide, correct? What was that one thing that was different in each of these tubes? The pH. So to get a low pH, we put in acid. To get a neutral pH, we put in water. To put a high pH, we put in base. All right, lastly, I noticed one more thing on here. Do you see my little pink bubbles? Yes. Again, my bubbles are indicating catalase is working, so that should go with your data, that it only worked in the test tube that had water because the pH was near neutral on that test tube. So I just want to explain the drawing that I did. Again, all of that information is on table 6.2. I just find sometimes having a visual to go with it helps us out. It's okay. All right, moving ahead. Chapter 6 is photosynthesis. So let's go on to page 67, photosynthesis. And I found this chemical reaction on page 67. You probably already boxed it or highlighted it. This is page 67 in your laboratory manual, page 67. But I went ahead and wrote it down here. CO2 plus water. They put solar energy. I couldn't fit that all in, so I summarized it with the word sun. Everybody's with me? Okay. And then this is CH2O. CH2O. Does anybody know what we call CH2O in everyday life? Carbohydrates. Good job. That's carbohydrates. CH2O is carbohydrates. And then everybody knows O2 is oxygen. Okay, so here's what it says. Know the reactants and know the products of photosynthesis. So what are the reactants of photosynthesis?
Carbon dioxide, water.
Carbon dioxide and water. Everybody agree?
Yes.
What are the products of photosynthesis?
Carbs and oxygen.
Carbs and oxygen. Carbohydrates and oxygen. It's okay on knowing the reactants and the products, because you're going to have to. Okay, it's like the basics of photosynthesis right here. Okay.
You want us to write like the, what was the first one again?
Okay, Jordan, go ahead.
So you guys want us to write the equation or just like the words in there?
No, you have to apply it. Like if I told you, okay, I got this plant and it's photosynthesizing, what gas is it using? So what's the answer? I got this plant, it's photosynthesizing, what gas is it using? Carbon dioxide. I got this plant, it's photosynthesizing, what gas is it producing? Oxygen. Now, speaking of the products, do plants do photosynthesis to make oxygen? No. What are they doing it for? To make carbohydrates, glucose. That's very good, Kaylin. To make glucose. Okay, so that's why I put a star right there. That's why plants are doing this. They don't really care about making us oxygen. That's not like, oh, I'm going to do photosynthesis so the humans could breathe today. Okay, they don't even care. They're making carbohydrates for themselves. Is everybody okay with photosynthesis, the reactants and products? Yes, ma'am.
What were the first ones? CO2. What's it called?
Right here, CO2. I'm sorry?
Yeah.
CO2 plus water. Those are the reactants. We can bracket those and put an R right there. And then these over here are the products. Yes, ma'am. Anybody else? Okay, great. The next question was, what organelle does this process occur? Still looking on page 67. You should know that this happens in the chloroplast. This is a very simple question, I think. Sometimes in this class you guys get caught up in the details and you forget the big picture. Like you probably know the light reactions occur in the thylakoids and the Calvin cycle occurs in the stroma, but you may forget the whole thing occurs in the chloroplasts. So this is just a big ticket question, where does this occur? All right, and there's a nice picture of this on page 67. You should be able to recognize a picture of a chloroplast. It's okay on that one. Super. All right, next page, page 68, paper chromatography. Paper chromatography. So paper chromatography, everybody, was a separation procedure. Let me remind you of what we did. We took a leaf and a piece of white paper, and you mashed the leaf a minimum of 10 times to get the pigments out of the leaf onto the paper. Do you guys remember doing that? Yeah. And then you can look at the pictures on page 68 to kind of remind you what we did. We put it on it was really a paper clip. They have it labeled as a hook, but it was a paper clip. And it was on a cork stopper, and we lowered it down into a test tube that had a solvent. So the question is, what kind of solvent was it? And the solvent was nonpolar. I'm not going to ask you chemically what it was, but I am going to ask you what kind of chemical family the solvent is in, and the answer is nonpolar. You remember nonpolar means it didn't have charge. Just a side note, I won't ask you this, but it was mostly ether. You probably heard of ether. Okay, so nonpolar solvents dissolve nonpolar things. And the reason we chose a nonpolar solvent is because the pigments of photosynthesis are also nonpolar. So that's why I chose a nonpolar solvent. And then I found how does it work on page 68 also. Handwritten on page 68. Check it out. I think it's in your lab manual if you were here that day. Did you write it down? There it is. Paper chromatography separates molecules. How? Based on their solubility. That means how well they dissolve in the nonpolar solvent. Now, you do have to know the pigments. Page 68, letter C. Put a star on that one. Page 68, letter C. Don't forget it is a multiple choice test. But you are going to have to know the top band is carotene. The next band is xanthophyll. The next band is, what is the next band? Chlorophyll A, and the next band is chlorophyll B. And I just, instead of taking a picture of that, I just drew my own up here. But there is this last question up here. Which of the four is most nonpolar? And these are your four choices. Which of these four are the most nonpolar? So,
The most important is carotene.
it is. Deanna's right. It's carotene. It's carotene. This one's the most nonpolar. Deanna, can you comment on how you knew that correctly?
It's like, it's something with the color.
Nope.
No, it's the furthest away.
It's the furthest away. It moved the furthest. That's good. Good job, Team Table 3. Working together. Okay, so because it moved the farthest, it must have dissolved the best in the solvent. So carotene is the most nonpolar. Try this question. Who's the least nonpolar?
Chlorophyll.
This one would be chlorophyll B. So this would be the most nonpolar, and the bottom one would be the least, because it hardly moved at all. So it didn't dissolve very well at all. It hardly moved at all. Is everybody okay on that question? Super. Okay, on our way to page 72, I want to stop by page 70. Everybody turn to page 70, stop by on page 70. Okay, what are we trying to show with figure 6.3? Okay, look at figure 6.3. What is this visual trying to show you? White light is... I'll keep us going. White light is refracted into all the colors of the rainbow. Aren't those the colors of the rainbow? It looks like a Skittles commercial. Okay, it's the colors of the rainbow. You've seen this before with glass prisms, or even if you just spraying water through a water hose. Haven't you seen the light refract into the colors of the rainbow? Anybody ever seen a rainbow in the sky? Me too. Yeah, me too. And so that's the refraction of white light as it's passing through all of the water in our atmosphere. Have you noticed when rainbows show up? After it rains, when there's so much moisture in the atmosphere, it's the water in the atmosphere refracts the light into its components. So page 70 is trying to show you white light is made of all the colors of the rainbow. You knew that. I'm just pointing it out to you. Okay. Remember, today's supposed to be a review, which means you already know this stuff. I'm just pointing it out to you. Okay, we were on our way to page 72. Please, everybody, join me on page 72. I have studied the absorption spectrum on page 72. Now, I'm going to change this question just a little bit. I want to add this letter right there. Be more specific. What did I add? A. Because if you look on page 72, there are different colors of chlorophyll. There's chlorophyll A and chlorophyll B. So let's all just zoom in on chlorophyll A. Look at that absorption. Do you agree with what I wrote down, that chlorophyll A absorbs the best in purple, also known as violet, and then it comes on down a little bit there in blue, and then it pops back up in orange, and it has a really nice peak in orange, and then it's starting to come down in red. Everybody okay with those colors? So really, it's really strong in purple and orange for sure, but it has some absorbance in blue and red. Everybody's okay on that one? Okay, how about the least? Green and yellow. Okay, so the question down at the bottom is, how do you know? Okay, how do you know what it's absorbing the best? What are you looking for?
The absorption.
That's absorption. So do you look for peaks or valleys? Peaks. Yeah, we're looking for the peaks. We're looking for peaks here. And then what are you looking for what it doesn't absorb? The dips. The valleys. The dips or the valleys. So you're looking for peaks to know what absorbs the best, and you're looking for the dips or the valleys to know what it's not absorbing. Is everybody okay with that one? All right, everybody turn, look out the window. Everybody, look out the window. I see plants out there. Do you see plants? What color are they?
Green.
Green. Why?
Because
Because they...
they're not absorbing the
They're not absorbing green. If you're not absorbing, you're reflecting. So what you see is not being absorbed. What you see is being reflected. Is everybody okay with that? Okay, moving on. Okay, so plants have more than just chlorophyll in their leaves. We've looked at this question several times before. Why is it beneficial for plants to have several pigments in their leaves to do photosynthesis? And I wrote, it helps them absorb more colors of light. Thus, they can absorb more, what's the capital E stand for? Energy. Is light energy? Yes. And again, page 72 is a good place to reference, to look at the idea of a plant having multiple pigments absorbing in multiple wavelengths, thus they absorb more energy. Are you okay on the idea of why plants have multiple types of pigments? Super. Okay, so I'm going to ask you the next question. And this is still on number 4. Still in the second part of number 4. Given your answer, what color of light is the best color of light for photosynthesis? White. And I agree with Jordan. Why is white light the best color of light for photosynthesis? It's all the colors. Are you okay on white? Okay, so what color of light would be the poorest color of light, most inefficient color of light for photosynthesis?
Black.
No, black is a no.
Green.
Green. Look out the window. It's green. You can tell by looking out the window. Green is not the color of light you want to use for photosynthesis. How can I tell by looking out the window? Because plants don't absorb green. What do they do with green? Reflect it. So green is the worst. So I wrote that down for you. What color of light is the best? It's white. What is the worst? It's green. Let me chat real fast about black. There are no such thing as black lights. Okay, what you and I might call black light is ultraviolet. You seen those lights people call black lights? Yeah, that's ultraviolet light. You guys have seen it. It's glowing purple, isn't it? Yeah, that's ultraviolet light. Black is the absence of reflection of light. Okay, so I'm looking at Kaylin's shirt today. Let me see who else. Oh, and Deanna's shirt, Eric's shirt. I don't know, Denise, is that navy or black? It's black, okay. Black up here. Okay, so black pigment, is it reflecting any light? No, it's absorbing all the colors. Okay, it's good. It's absorbing all the colors, so it mixes in. Maybe you did that when you were a kid. I don't know. Here's what I used to do. We used to have these paints in these strips, and you buy them, I don't know, watercolors? That's what they were called. And you remember you would like do yellow first, and you put it in your little cup of water and wash it off, and then you want to do red, and you do it, and you put it back in your cup of water and wash it off. And by the time you got finished, what color was that cup of water? Black. It's black, because it was all the colors mixed together. That's good. See, you knew it. Okay. Everybody's okay on that question? We're going to number 5. Number 5. It's a true or false question. Most plants carry out photosynthesis only during the day. And that is true. But I want to know why. Okay, tell me again, Deanna.
Something's open like this.
The stomata open during the day. And then what else happened? Look outside. What else is happening during the day? The sun, yes. We call the time period when the sun is out, we call it the day. Yeah. Yeah, I'm glad we had this chat. Okay, and so I underlined the word the part photo and photosynthesis up here. Did you guys notice that? What does photo mean? Like photograph? Photosynthesis? Anybody know what that means? It means light. Who told me that? Charlie? Good job, Charlie. Photosynthesis means light. You've got to have light to do photosynthesis. Charlie's right. So that's why I underlined the photo of photosynthesis there. You've got to have light to do photosynthesis. Okay, then Deanna's right. You got to open up those holes during the day so that the CO2 can come in. Very good, Deanna. Okay, check out this next one. True or false? Plants carry out cellular respiration only at night. Why?
Because there's no sunlight to make photosynthesis.
No. Nothing to do with sunlight. What's cellular respiration trying to make? ATP. Cellular respiration tries to make ATP. When do cells need ATP?
During the day. Always.
Always. That's great. They need it always. Okay, so the reason this is false is plants carry out cellular respiration 24/7. What does that mean, 24/7?
All the time.
All the time. All living cells carry out cellular respiration 24/7. I don't care who you are. Animal cells, plant cells, bacteria, fungi, protists. It doesn't matter who you are. All cells carry out cellular respiration all the time. So that's what makes this false. Everybody's okay on that one? Super. Next question, number 6. Is that number 6? Yes. Okay, guys, I found number 6 on page 74. Page 74. I think we took some notes. Everybody turn to page 74. Take a look at your notes. I have, why did the phenol red turn yellow when carbon dioxide was added?
Because plants didn't do photosynthesis.
You're ahead of me. Okay. Because the CO2 was high. Now, let me tell you what we did, because you maybe forgot. What is she even talking about right now? I'm talking about one day we took a beaker and put water in it, and then we put this stuff called phenol red. Then I handed somebody on your table a straw. You may remember who that was? Okay, yeah. Okay, it was Diamond. So you kind of remember who it was on your table. And then the person with the straw, what did they have to do? They had to blow into the water. So check it out. Here's the water that was in their test tube. And then what was in their breath? CO2. So there's the CO2 from their breath. And look, it added together and made carbonic acid. So the pH indicator phenol red turned yellow because the pH became acidic. We made acid in there. We made acid. So the answer is we made acid with the CO2 blowing into the water. Now, when we first did this chemical reaction, we were going this way. Everybody check at my arrow. Because you were pushing that CO2 in so hard, we were always going this way. But then when you stop blowing and the beaker has no more CO2 just rushing in, then the reaction can start going back the other way. So this is a reversible reaction. When you're blowing the CO2 in, it's all driving forward to the right, but then once you stopped, then the reaction could go back to the left. Now the reason I mention that is what you did was you added lots of CO2 to the water. So you wrote down, I think on page 74, that when the solution is yellow, that's high CO2, and when the solution is red or pink, that means low CO2. Did you guys write that down? Okay, then the next day. Don't you like my drawing, huh? Check it out. I got the sun right there. See, there's my sun. Let me color my sun. It's not even colored. There. That's better. There's the sun. There's my beaker. There's my plant. I put a little plant in it. You guys see my plant? Okay. And I put this one in the light, and I came back 5 days later. Check out your notes, page 74. When we came back 5 days later, the beaker we left in the light should have been back to pink. Is that true? And I want to know why. So I wrote down that the plant did photosynthesis. And here's the catch, though. It did photosynthesis using up the CO2. So if you use up the CO2, then the reaction goes the other way and your pH goes back to neutral. So it went back to a neutral pH. You used up all the acid is another way to think about it. Now all the acid is gone And now you're just back to where you were at the beginning. Everybody's okay on that one? Okay, now look at your dark information. We also put a beaker in the dark. When we came back after 5 days, the beaker in the dark, did it turn back to pink? No, what color? Yellow. Why did it stay yellow? Because there was no light, it did not do photosynthesis. Okay, so there was no, so here's mine. That's the moon, right there. Can you tell? Right there. Why did I put a moon there? For the darkness. I should have drawn a drawer, but you can tell I'm not really big into art. Okay.
So I have written down that CO2 stays high and it's due to cellular respiration.
That's right. Now, let us be clear, Haley. This plant was doing cellular respiration too, but not a whole lot. It was doing more photosynthesis. This plant was doing cellular respiration also, but no photosynthesis. Okay, that's good. But okay, no light, no photosynthesis? Okay, and we learned that if it's yellow, that means that we have lots of CO2 in there. And if it's pink, that means we have low CO2. That should go with your notes. Everybody's okay on that question?
Say that again.
High CO2, pink is low CO2.
Yeah, I'll go back. There's my pink is low CO2. Yep. Anybody else on number 6?
No, wait. The light pink is high CO2, not the light. Like it would be in the light is low CO2.
Pink,
Will it always be yellow or pink?
yes.
Thank you. Okay.
Pink means low CO2. Yellow means high CO2. Is that better?
Thank you.
You're welcome. All right, I like question number. Everybody look at question number 7. And look at the visual on page 74 to go question number 7. We're going to question number 7. Why is it important to have autotrophs, heterotrophs, and decomposers in an ecosystem? Now, this is page 74, figure 6.6. Everybody take a look at that picture real fast. So, I was looking at that picture, and I started thinking about how I have something like that picture in this lab. I have that little aquarium back there. You guys ever seen it? It's back behind Samaria. Yep. Miss Samari, tell me if I'm telling the truth. Are there some orange fish in there? Yeah. Is there a green plant in there?
Yes.
Are there rocks on the bottom?
Yes.
And there's water? Yes. You know what else there is I'm going to draw? Mr. Sunshine. Everybody look at the window. Isn't there a Mr. Sunshine involved here? Okay, watch me draw Mr. Sunshine. There he is. Okay, I got myself a little ecosystem. Let's see. Who are the autotrophs? The plants. Everybody okay on that one? Who are the heterotrophs? The fish. And where are the decomposers? Tell me again. You know what? If I had algae in there, which I work real hard not to, they would still be autotrophs. Algae do photosynthesis. Anybody else? I'm looking for the decomposers. Ideas of they're there. Where are they? They're on the rocks. Kaylin is right. They're on the rocks. That's why you put rocks or gravel in an aquarium, to provide a surface for your decomposers. That's why you put it in there. I don't know if you knew why you put rocks on the bottom and why you usually use little rocks and not big boulders to give lots of surface area for your decomposers to live on. Now, who are these decomposers living on the rocks? Bacteria. The decomposers are the bacteria. Can you tell I have rocks on the bottom? Okay, just checking. Again, I don't do art well. So, why are each needed? Let's talk about the autotrophs first. Why are the autotrophs needed in my little aquarium? To make oxygen. That's good. So I'm going to make some oxygen. Oopsie. Don't know what I did. Oh, wait a minute. Sorry. We'll go back to this one. I want to go back. There I am. Oxygen. What else do the plants make? Food. Do you guys mind if I use CH2O to stand for food? What does CH2O stand for? Carbohydrates, and then, the plant makes amino acids and proteins and lipids from it. Okay. Now, what do the fish do with the oxygen? They respire it. What do they do with the carbohydrates? They eat it. And then they make something. Make two things. If you don't know, take a look at the picture, page 74. What do the fish make? Carbon dioxide and water. And who needs the carbon dioxide? The plant. And who needs the water? The plant for photosynthesis. Does everybody see we got a little cycle going right there between our heterotrophs and our autotrophs? So heterotrophs need autotrophs for food and oxygen. Autotrophs need heterotrophs for CO2 and water. Are you okay with that? Ma'am?
So the heterotrophs are needed to make CO2 and water?
Mm-hmm.
How do the fish make water?
If
Like
you
by
release
okay.
Let's talk about what else fish make. What else do fish make besides pee? Poop. Now, in biology, we don't call it pee and poop. Okay, what do we call pee in biology? Urine. And what do we call poop in biology? Feces. Feces. Now, the plant is not going to use those waste products except for the water and CO2. That's all the plant wants is the water and the CO2. So what's going to happen to those other waste products? Who's going to take care of these things dropping out of these fish? The bacteria living on the rocks. Does everybody see why you have to have decomposers? To get rid of the wastes. Bacteria are used to get rid of the waste. Now look, guys, what if Miss Steinke forgot about her tank? Let's just say I went away for the Christmas holidays. I was gone for, I don't know if you know, I think this Christmas holidays, we're going to be gone for 4 weeks. You're going to be gone like 5 or 6 weeks. I forgot about my tank. Let's just say it happened. And my fish died. What is going to get rid of my fish in the tank? The bacteria. So everybody see, you've got to have decomposers to get rid of dead stuff and waste products. Are you okay on why you have to have decomposers? All right, now, Samari, would you go look at that tank and tell me, is it completely closed up? Because from right here, it looks like that top completely encloses that aquarium. Is that true? There's no openings at all. So there is an opening. Okay, so she said there's a waterfall on the back. If you want to look later, go ahead. So the top is not completely closed. There is an opening. Now, what if the top was completely closed?
They could not.
They don't have no oxygen.
Oh, hold on. I disagree. There would still be some oxygen coming from where? The plants. That's right. But would the tank last as long? Would the fish and the plants last as long if I put a lid on it?
No.
No. Everybody agreed that we would limit the amount of gases made if we put a lid on it. If the lid is open, then gases can diffuse in and gases can diffuse out. So you may notice most aquariums are not completely sealed so that gases can diffuse in and gases can diffuse out. But theoretically, if you have everything balanced, it should be able to make it. The balance is the problem.
So is that why goldfish die quick? Because if you close the tank?
Let me tell you, goldfish urinate and defecate a lot. Okay, and so why they die fast is their waste products really add up. And most people just put goldfish in a tank with no gravel on the bottom and no aerator. So they're just dooming their fish. So poor little goldfish, they die swimming around in their urine and feces. Well, I'm just telling you what happens. I'm just telling you what happens. Okay, so now we're moving along. We are moving to chapter laboratory 7 on cellular respiration, page 77. And I have know the reactants and products of aerobic and of anaerobic cellular respiration. Now, of aerobic cellular respiration, I wrote it down for you. I found this on page 77. Now, guys, I want you to notice that I also added in how many ATP this thing makes. Everybody see I added it in? How many ATP can you get in aerobic cellular respiration? 38. How do you know this thing is aerobic? Oxygen. So you might want to circle that part or make note of that part. I just wrote aerobic and then shot an arrow up to that oxygen. That's how I know it's aerobic. Okay, don't worry that you have to write this reaction. It's a multiple choice test. You have to be able to spot it, pick it out. Okay, of a list of reactions, you should be able to check this one as being aerobic cellular respiration. Okay, this one is anaerobic, and I found it on page 78. It's at the top of page 78. Now this one is actually alcohol fermentation. This C2H5OH, that's alcohol. You may remember we use alcohol fermentation to make beer, wine, any kind of liquor. And we also use alcohol fermentation to make bread rise because of the CO2. Yeah. The cells that like to do alcohol fermentation are yeasts. So you may remember yeasts are used in beer making or wine making or alcohol making or making breads. Sorry, I'm just playing. Just playing. Let's just check out some questions. And I would never do this with a full bottle, but let's say I had an empty bottle of beer on the test, because we are not allowed to have alcoholic beverages on the campus of San Jacinto College. So let me be clear. I would have emptied it off campus. But I have an empty bottle of beer on the test, and I ask you what chemical reaction is used to make this product. You pick this one, right? Right? All right, so let's just choose between this chemical reaction and the one previously. Does everybody remember the one previously? Between this one and this one. We'll call this one A, aerobic. We'll call this one B, anaerobic. Which one are you doing right now? A. You're doing aerobic cellular respiration. Everybody agree? Okay. Let's just say I put a loaf of bread on the test and said, what happened here to make this loaf of bread? Would it be A, aerobic, or B, anaerobic? B, anaerobic. Okay. Pretty much we use anaerobic fermentation to make lots of foods. Very good. Nice summary there. Okay, this was an easy question. I found this one super easy. Number 2, both anaerobic and aerobic cellular respiration ordinarily begin with what molecule? Everybody check it out. They both start with glucose? Yep. That's what I found. That's an easy one. Question number 3, in which reaction is the glucose completely broken down? Any guesses on that? You can go A, aerobic, or B, anaerobic. You think it's B? Yeah. Think it's A? Okay, let me tell you how you would know. It depends on how much energy, ATP, you get out of each. Remember, you eat glucose for a reason, for energy. Don't you have glucose in your diet for energy?
Yes.
Yeah, you're not building your hair with it. You're not building your muscles with glucose. You're eating glucose for energy. So which one do you think breaks the glucose completely down?
Anaerobic.
Nope, you only get 2 ATP out of it that way. It's definitely aerobic. How can you know it's definitely aerobic that's breaking it down? You get 38 ATP. You get way more energy out of it, so you must be completely breaking it down. So the answer is aerobic.
Will you ask that?
Yes.
Why are we... Yes.
Yes.
Gee.
Okay, help me out. Let's say the aerobic cellular respiration was the letter A. We were calling it chemical reaction A, and anaerobic was chemical reaction B. Without looking, can you tell me how many ATP the aerobic cellular respiration makes? 38. And anaerobic? 2. So you got that down. Very good. Got that one down. But okay, on question number 3. Okay, question number 4 is just to remind you what it looked like. Okay, so we did the fermentation experiment, and our data is on page 82. We set up 4 test tubes, and I kind of drew what they look like. These were where we were using itty-bitty test tubes. And look at your data, page 82. Test tube 1 had glucose. Test tube 2 had fructose. Test tube 3 had sucrose. Test tube 4 had water. And I just wrote it down for you up here. And then test tube 1, 2, 3, and 4 all got yeast. And you may remember we had to fill those itty-bitty tubes to the tippity-top, and then we had to put it inside of another test tube, and then what did we do? Flipped it. Okay, we put them over in that incubator, and a few minutes later we came back, and we noticed that some of them had gas. Just putting dots in here. Gas. That was our fermentation experiment. So you guys do remember doing that? Yes, okay. All right, so here's the questions that go with it. Did the gas bubble increase or decrease with each of the solutions used? I checked our class data on this. We should have class data, page 82. I think we had an increase in the bubble in test tubes 1, 2, and 3. Yes. I think 1 was the best. Is that true?
Yes.
2 came in second place?
Correct.
And 3 came in third place?
They were tied 3 and 4.
3 and 4 tied?
No, 2 and 3.
I mean 2 and 3.
2 and 3 tied? Okay, so first place was glucose. Second and third place tied were fructose and sucrose.
Yes.
And dead last was water.
Yes.
Okay, how come water didn't make the gas? Can yeast cells use water to do cellular respiration? No. Can any cell use water to do cellular respiration? No. So I wrote that down. Cells cannot use water for energy. They can't do cellular respiration with water. What was the purpose of test tube number 4?
Control.
Yes, that was our negative control. Okay, guys, most importantly, you have to know what was the name of that gas. So let me star this. What was the gas we were measuring? CO2. Because the cells produce cellular respiration. You have to know the gas cells make in cellular respiration. It's called CO2. Everybody's okay? Okay, I couldn't draw this. I tried. It just looked ridiculous. So this is number 5. This is the visual that goes with number 5. Sorry, you're just going to have a look at it. This is what we did on number 5. You remember setting these up? Yes? Okay. And if you want a page number to look for number 5, page 79 looks real good with number 5. We set up 3 test tubes, cotton in the bottom and a CO2 absorber called KOH. Test tube 1 got germinating peas. Test tube 2 got dry peas with glass beads. Test tube 3 got just glass beads. So again, Page 79, definitely want to look at page 79 for this. Page 79, look at the pictures. And then our data, everybody, our data is on page 80. So the setup is kind of on page 79, and then our data is on page 80. Let's look at our class data. Looking at just class data, which vial had the most movement of the shaving cream? Do we have class data? This is for number 5. Number 5. I'm sorry. I don't know if, did I even have number 5? Let me see it. Okay, so which of, if you just will glance at your book, page 80, which of the vials for class data had the greatest net change? I don't remember. One?
Wait, for the class data, one.
Everybody look at class data, because your individual data probably sucked. But class data always comes out looking nice, yeah. Okay, so class data, everybody agree one? Germinating. Okay, that meant that they were doing cellular respiration at the highest rate. Did number 2 come in second place?
Yes.
Okay, indicating the cells were still alive there, but they were not using up oxygen very fast. And I was hoping test tube number 3 came in last place.
It did.
Because last place would mean that they didn't do much cellular respiration, and that works out for me because there were no cells in that test tube. So if you don't have cellular respiration, you don't have cells doing cellular respiration, then that doesn't make sense that they can't do it. So glass beads should not have done much. Is that true for our class data? I think there was one group that had that one move. I'm just doing it off of memory. So did everybody understand that group number 3 or vial number 3 was our control? Okay, good. All right, so go into this question, which I think is number 6. Which direction did you expect the shaving cream in the pipette to move during aerobic cellular respiration? I wrote inward. If you don't like in, you could do inward. And why? Because the cells were using up oxygen doing aerobic cellular respiration. The cells were using up oxygen doing aerobic cellular respiration. Everybody's okay with that? All right, so we already answered the last question. Everybody look at number 7. What gas was taken up by the germinating peas doing aerobic cellular respiration? Oxygen. The answer is in the question. Everybody find how come the answer oxygen is in that question. The word aerobic. Very good. So oxygen is the answer to number 7. But notice the answer is in the question. It's in the question. Okay, on number 7 and exercise 7. All right, super. I think that takes us to exercise number 8. All right, so mitosis and meiosis should be still pretty familiar to you. We were just talking about it like last week. So I'm just going to kind of gloss over some things real fast, starting with the cell cycle, page 86. I'm going to kind of go fast here, page 86. So I just took a picture of a cell cycle. It looks pretty similar to the one you have on page 86, except I drew some stuff on it. So we're going to draw stuff on this. And so what we're going to draw is whether the DNA is single-stranded or double-stranded, and whether it is chromatin or chromosomes. Now, the only difference between my cell cycle and your cell cycle is that G0. Everybody see it on there? This one's actually a better cell cycle. It has a little more detail. But you can look at the one that's in your book. Everybody find G1, S, G2. Get yourself something to write with so you can draw on your cell cycle. We're going to start with G1. Okay, so we're on page 86. Everybody has a pencil in their hand. We're going to draw on our cell cycle. We're going to start with G1. And during G1, chromosomes are very long, and they're thin, and they're single-sided. So what I'm drawing in pink up here is a chromosome. Long, thin, single-sided. G1. Did you draw it on your G1? Single-sided. So check this one out. Now I'm going to go to S. Chromosomes are still long and thin, but now what? They're double-sided. If I look at my difference between G1 and S, how I drew my chromosomes. How come they're double-sided? How come they're double-sided? They replicated. You guys spotted it. During S, the DNA made a copy of itself. So now does everybody see why the chromosomes are double-sided? Okay, now during G2, they're still long and skinny, and they're still double-sided. No change. Now, everybody notice how it's long and thin during these purple parts. G1, S, and G2. Collectively, what do we call G1, S, and G2?
The prep stage.
Prepare, okay. Also known as interphase. There it is. So it's kind of long and spread out and hanging out all over the place during interphase. Everybody's okay with that? All right, during mitosis, the DNA coils up real tight. So notice when I come over to prophase, now I'm going to make it really coily and tight. It starts looking more like the letter X. You guys see that over here? Do you see that it's double-sided? So what's the difference between how you saw it in G2 and how you see it in prophase? What's the difference?
They're not long.
They're not long. Now they're all coiled up and tight. Are you okay with that? Okay. We're not doing pro-metaphase, so scratch it out. No pro-metaphase. Okay, metaphase. They still look like that. Double-sided. Nice and thick. Metaphase. But not in anaphase. Because something happens in anaphase. What happens? They pull apart. Okay, so pulls apart. How about in telophase? Still the same. Now check it out. We finished the circle. Check it out. Here it is, single-sided, all coiled and tight, and then we go back to G1, and now what is it? Single-sided and all spread out. Everybody see we got a cycle going here, don't we? But instead of one cell, now we have two. But we got this cycle going on. That's why we call it a cell cycle. A cell cycle. Is everybody okay on my drawings? Now, I have know the form of DNA in each stage. And these are going to be your two choices. So write this down. These are going to be your two choices. This goes with the second part of number 1. Know the form of DNA in each stage. Here's my two words. Chromatin and chromosomes. One of these is the DNA all spread out. Chromatin. Denise is right. The one that's all spread out, taking up all the space. You know, we drew it kind of long like that. That's chromatin. Chromosomes, they're not all spread out. What do they look like? They're the ones that are coiled up tight. When you drew them like this, the coiled up tight, those are chromosomes. See, they're the same thing, but one's all spread out and one's all packed up. Everybody okay on the difference between chromatin and chromosomes? Yes? Great. And don't forget, page 86 would be great for that. Page 86, where you drew on your cell cycle. There it is again. Okay, I did a really good drawing here. Man, I'm so proud. Of chromatids and centromeres. Page 86 is still the right page to be on for this. Everybody take a look. You labeled your chromatids and centromere. Chromatid, centromere. But I do have a quick question. Why did I draw two of them? And one of them's red and one of them's blue. Why did I do that? What are these two together called? Homologous. Where'd you get the red one from? Where'd you get the blue one from? Now, do we seriously call those mom chromosomes?
No.
What do we seriously call them? Maternal. And do we seriously call them dad chromosomes? Paternal. But that's why I drew two of them, because chromosomes are in pairs. One from your mommy, one from your daddy. You people don't even ever thank them, do you? Thanks for those chromosomes, Dad. You know, he didn't have to. It's the little things we need to thank people for. Okay, so you're okay on chromatid and centromere? We had those labeled on page 86.
What is it? Oh, okay.
Homologous. Sorry, homologous. Homologous. There's a lot of letter O, letter O, letter O in there. Okay. All right, guys, then I had a few other vocabulary words. Number 2, we know what a chromatid is. We know what a centromere is. We labeled them. And then there were just a few vocabulary words here. I went ahead and defined them in case you didn't know what they were. Cytokinesis is division of the cytoplasm. We usually associate cytokinesis with telophase. Spindle fibers are those proteins that separate the chromatids during anaphase. They pull them apart.
Proteins that what?
Pull sister chromatids apart. I got it up here. And then really the only thing I want to mention on centrioles is that every animal cell has one pair. You don't have to worry about their function because we don't really know what they're doing. We don't have any idea what they're doing. But they are in animal cells, not in plants. We're not exactly sure what they're for. So I put that they're in animals and there's one pair per cell. And then I'm just looking at that list. We already did chromatin and chromosomes, yes? Okay. But okay on that one? Okay, the difference between plants and animals on mitosis, I came up with these two things. This was my first difference right here, and this was my second difference. I figured out that animals use a cleavage furrow to do their cytokinesis, but that plants don't. Plants use a cell plate. You guys know that, right? Okay. And then I just restated what we just mentioned before, that animals have centrioles and plants do not. Can you do a cleavage furrow? Can I point at one on a slide and you be okay on that? What's a cleavage furrow look like? Right? What's a cell plate look like? A line. If you don't know these things, page 88 and 89 are valuable resources in your your lab manual. Page 88 and 89. Everybody's okay on this one? Look at this next question on page, number 3, second part. Explain why cleavage furrowing is a suitable mechanism for cytokinesis for animals, but not suitable for plants. How come? Because animals don't have a cell wall. What's the problem with having a cell wall? You can't squeeze in, right? So here's what I wrote. Well, first I drew it. Everybody's okay on my drawings? They're so simple. Okay, and I put that a plant's cell wall is rigid. So that's why plants cannot do cleavage furrows. Okay, then lastly on mitosis, I want to remind you I'm going to test you on mitosis using slides. So study page 88, 89. Models, which are over here on the side, and then the pop beads. Hey guys, we are running out of time, so I'm just going to dash real quickly over to meiosis. It's on the back. Just have a couple things to tell you about. Most of this you can find in your lecture notes. And I'm just going to keep talking to you. And if you need to go, then feel free to go. But I did do this work for you, so I wanted to share it with you. If you want to go, feel free to go. See you guys. I put that diploid is where you have 2 sets of chromosomes. And that's true for most cells. They're diploid. What does this little 2N at the top mean? That's our symbol for diploid. Does everybody know DI means 2? Yes? DI means 2? Colton, you're okay? Jumped off and hurt your knee? Or he's dancing. He's dancing. Whereas haploid means 1 set of chromosomes. And I'm just again circling the term that means haploid. Hey guys, after if you had to pick between diploid and haploid, what do you think most of your cells are? Diploid. Tell me cells in your body that are haploid.
2.
23.
Yeah, they have 23 chromosomes. Where are they?
Oh, sex cells.
They're your sex cells. So ladies, you have them in your ovaries. Gentlemen, you have them in your sperm. Is everybody okay with that? That most of your body's diploid, but your sex cells are haploid. You're okay with that? And then Alex is right. Our diploid number is 46. For humans, it's 46. But for our sex cells, for humans, it's only 23. Okay, and then I identified homologous chromosomes as a pair of chromosomes that carry information for the same traits. You were telling me one came from mom and one came from dad. So that's a real quick repeat. Okay. That was to remind me to tell you to look at page 96 and 97 for the stages of meiosis by Jasmine.
What page is 93?
It was 96 and 97. That picture right there is from page 96 and 97, stages of meiosis. So take a look at that. And then let's head to page, we're going to finish that real fast, page 101. If you'll glance at page 101, like, Ms. Steinke, what kind of questions will you ask me? I'll say, hey, where does spermatogenesis occur inside of a human? In the testes. Everybody hear Deanna? Spermatogenesis occurs in the testes? Okay, try this question. Per original diploid cell that does meiosis in the testes, how many functional sperm can you get? 4. That's good. Is that Jeannie? Michelle? Nobody? How did I get girls out of that? Yeah, it was so Eric. The answer is 4. Take a look at the picture top page of on the top of page 101. Let me repeat the question. Per original cell that does meiosis in the testes of a male, how many functional sperm can you get? The answer is 4. They look at the picture. Try this question. Per original cell that does oogenesis, how many functional eggs can you get? 1. What organ in a female does oogenesis? Ovaries. Now, guys, are we going to have to be able to tell an ovary from a testy on the test? Yes. Page 102 and page 103. Make sure and study those. I just mentioned some of the parts you have to study there. Page 102 is the ovary. Page 103 is the testis. So make sure and look at that. Stages of meiosis, page 96 and 97 for our pop bead chromosomes. And then lastly, you have to know what synapsis crossing over is. Synapsis and crossing over. I have that synapsis is when homologous chromosomes wrap around each other, and they do that in prophase 1. Crossing over is where they switch information. Does anybody know why cells do crossing over? Variation. Very good. Variation. Okay, so for number 5, I want you to go back and take a look at page 96 and 97. Here's synapsis. Everybody okay on synapsis? I'm going to just go ahead and pull this last one up. Crossing over.
YouTube, right?
It's going to be on YouTube. It's going to be on YouTube. Side note, guys, when you go down into Mendelian genetics, you do need to know that vocabulary there. We did all of that today in lecture. So when you're reviewing exercise 9, go to what we talked about at the beginning of lecture today. We were working those chi-square. We were working those 4 problems, getting our ratios. Mostly that was all for Exercise 9, Mendelian Genetics, our lecture today. Do you have to know those 4 ratios? Yes. Memorize the 4 ratios. Do you need a calculator? Why do you need a calculator for lab? To do the problems. What are the problems called? Chi-square. Very good.